All right, perfect. So we're going to start talking about the skin today. And lecture and lab will cross over. We'll hit a lot of fine points. So keep in mind, there's going to be some beneficial, I don't want to say redundancies, but some repetition. And so don't worry if you don't quite catch it in either lecture or lab, because most likely we will go over it again in the other part of the course. Meaning, if we go over something in lab and you didn't really quite understand it, no worries, no worries. We'll hit it again in lecture. You'll be okay. All right, for the most part. So we're going to start off with just going to kind of scratch the surface here and talk about the integument system. So first of all, when you hear this term integument, we are definitely talking about the skin. The largest organ in the body, right? You can see by looking at this picture, and I can zoom in a little bit here. And just this one section here, just to kind of give you an appreciation, just look at all of this stuff in the skin here, right? And we've talked about what you heard me mentioning when we were going through the different types of tissue in lab. There's epithelial tissue, connective tissue, nervous tissue, and muscle tissue, all right? And so certain organs will have all those tissues in it. Skin happens to be one of those. And so if you look here, we're going to go through a bunch of this stuff here and in quite a bit of detail. But I want you to kind of get an appreciation when we're talking about the skin and what it's made up of. So we're going to go through the different layers, the epidermis and the dermis, and then we'll break those down into their sublayers, and we'll work our way through. So when we're talking about the layers of the skin, let me go back real quick. All right, we have an outer surface layer, the epidermis, and then a deeper layer called the dermis. And we break down the dermis into these two layers, the papillary layer and the reticular layer. So today, we're going to start off talking about the epidermis. And that's the outer layer for the skin, and it's made up of epithelial tissue. Epithelial tissue. Okay, easy enough. So the specific type of epithelial tissue that we're going to see in this layer is called keratinized stratified squamous epithelium. Now, there's a term that I've used before with you folks when you've heard the word keratinized, and that is, okay, keratin kills. It does. Keratin is a protein that it helps to make the cell stronger, but in doing so, it will kill the cell. So when we're talking about the epidermis, we subdivide the epidermis down into 5 layers or strata. So number 1, this first guy here, stratum basale, is the deepest, and then the stratum corneum is the most superficial. So we're going to start off with the deepest layer, and we're going to work our way up towards the surface of the skin. Now, the correct pronunciation for number 1 is the stratum basale. I say basale, so when you spell it on the test, you don't forget that there's an E on there. And I'll do that with some of the words that I will use for you. Yes, I know that sometimes it's grammatically it's incorrect, but I'd rather have you spell it and get it right. So when we talk about these first 3 layers here, we're going to be dealing with our living cells, the keratinocytes, all right, but eventually they're going to die off and we'll be left with the dead keratinocytes. So let's start off with the first layer here, and that is going to be the stratum basale. And the stratum basale is made up of, all right, a layer of cells, one single layer, and they appear to be cuboidal or what we call low columnar cells. And so when we say cuboidal, we know that they're either going to look like this, they're as tall as they are wide, or low columnar, which they are slightly taller than they are wide. But keep in mind, when we're dealing with stratified epithelial tissue, who cares what the bottom layers look like? Who cares? When we're going to name the shape of the cell, when we're classifying our epithelium, and if it's stratified epithelium, it's always the apical layer, the topmost layer. So even though in the strata basal, the bottom layer, those cells will look cuboidal or low columnar, right? They are still, that epithelium is still squamous epithelium. So there's 3 cell types that we find in this single layer. The predominant cell type, which is the keratinocytes. Then we have the melanocytes, really cool cell. We'll talk about that in a moment. And tactile cells, touch cells. So the keratinocytes, these are the ones that are going to make our keratin. And that's the protein that is going to make our epidermis nice and strong. So these keratinocytes are in that basal layer there, that bottom layer. And their job is to just divide. And that's what they do. And so as you remember, when we talked about in lecture, we were talking about mitosis. When one cell divides, it leaves 2 cells. And so one of those cells, one of those daughter cells, is going to remain in the stratum basal. The other daughter cell is going to move into the next layer. That's how it works. Okay, so these keratinocytes in this layer are basically stem cells. They're just going to keep dividing, making new cells. And these cells, their job is to make that keratin, which will eventually kill them, but that's okay, because in their death, they have done us a great service. They have strengthened our epidermis. The second type of cell type is the melanocytes. I love these folks. They're wonderful cells, right? They're scattered throughout this layer here. And so they make this awesome pigment called melanin. And melanin's job is to protect the DNA from ultraviolet radiation, both UVA and UVB radiation. And so the actual cellular organelle that makes the melanin, we call those melanosomes. All right, and so they'll transfer the melanin to other cells in the area, mainly the keratinocytes. And those keratinocytes will then just slather that melanin all over their nucleus. And it protects the nucleus, all right, from that ultraviolet radiation. Because otherwise, that ultraviolet radiation gets in the nucleus. What's in the nucleus? DNA. And it can cause splicing of the DNA and create what we call dimers. And that's bad news, folks. Bad news. Certain genetic conditions can arise from that. You don't want to mess with that. All right. So nature has given us this melanin, right, to help us. And that melanin will give our skin darker pigmentation. Now, unfortunately for folks like myself, my melanocytes, all right, aren't as active as folks that have much darker skin than I do. All of us have the same number of melanocytes. I'm not saying we all have like 10,000, all right, but it's pretty much the same. It's just that some people's melanocytes are more active than others, hence the darker pigmentation. All right, the third type of cell that we're going to find in the stratum basal are our tactile cells, also known as Merkel cells. All right, these cells are touch sensitive. So when you actually apply pressure to these cells, they release these chemicals. And in the nearby vicinity, we have these receptors, free nerve endings. All right, and these chemicals will stimulate those nerve endings. And then those receptors will then transmit sensory information on their sensory neurons to your nervous system. And then therefore you will realize, oh, I'm touching something or something's touching me. Those are the tactile or Merkel cells. So those are the three cell types that you will find in the stratum basale. Okay, working our way superficially towards the surface of our skin, the next layer is the stratum spinosum. Now, this is the layer that just received a daughter cell, all right, or many daughter cells from the deeper cell layer. And so these cells have just finished dividing. And so once they've done that, right, now we're going to start to kind of weave our epidermis together. So it's in this layer where these cells start to develop connections to one another, right? Our intracellular junctions and our desmosomes. And these are going to be, all right, structures that are on the side of the cell, all right, and on the bottom of the cell. And I guess thereby on the top of the cell too, because one cell's bottom is another cell's top, in which now these cells start to pack themselves tightly in with one another, and they don't want things getting in between them. So they start to form these intracellular junctions in these desmosomes. Also in this layer, we have a really kind of cool cell called a dendritic cell, also known as a Langerhans cell. And these are immune type of cells. They can initiate the immune response. If they come across, all right, maybe a neoplasia or a dysplasia, some abnormal cell growth, or something that doesn't look quite right, these cells can initiate the immune response, and we can destroy those cells with that tissue, hopefully. And in fact, quite a lot of skin cancer that could become an issue, these cells intercept those cancerous cells usually pretty early on. They're really cool cells, dendritic cells. You'll learn more about them in the immune chapter. Our third layer of cells in the epidermis, all right, that layer is called the stratum granulosum. And it's here where our story of the keratinocytes takes a sad turn. They start to die. And it's kind of sad. Okay, it's kind of sad that this is happening because, yes, they do die. The cell starts to fill up with the keratin, and the keratin starts to impinge upon the space there for the organelles and in the nucleus, and those start to deteriorate and disintegrate. And so therefore, when all that happens, right, those cells are dead or dying. Right, not a very thick layer here, right? Only 3 to 5, or not a very strict stratum, all right? Only 3 to 5 cell layers thick. Then as our next layer is the stratum lucidum. This layer is really interesting because it's only present in thick skin, which is only going to be found on the palms of your hands and the soles of your feet. So I would mark this down as a very important concept, okay? That this layer, this stratum lucidum, not a very thick layer, only 2 to 3 cell layers thick, is only found in thick skin, palms of your hands and the soles of your feet. And then finally, our outermost layer, the stratum corneum. It's the most superficial layer here. This is the thick layer, anywhere between 20 to 30 cell layers thick, made up of dead keratinized cells that are tightly packed into one another. So because of these characteristics, right, this cell layer is going to have a dry appearance to it, okay? Which is good because a dry, what I'm looking for, a dry disposition makes it harder for certain microbes to thrive on. All right, fungus likes a moist environment, but a dry, thick area makes it tough for infection to occur, and it helps with abrasive forces acting upon it. So you can see here when we're talking about our skin, all right, we'll see this one single cell layer. There's the stratum basale, and it's got its melanocytes here, and it's also got its tactile cells here, mainly made up of the keratinocytes. And then our stratum spinosum, pretty much made up of daughter cells that came from the stratum basale, and our dendritic cells, these fellows right here, those are the Langerhans cells there. And then we have our stratum granulosum. This is where the keratinocytes start to produce, all right, that keratin. And then our thin layer of the stratum lucidum, only found in the thick skin. And then our stratum corneum on top. So this is pretty much considered to be thick skin right here, because you have, I won't even attempt to count the cell layers here, but it's many, many cell layers thick. Thin skin looks a little bit different. I'll show you. I've got a slide that contrasts that. So when we're looking at the epidermis, there's three things that we're going to notice with some of the variations in the epidermis. One is going to be the thickness, and I just talked about that. I'm going to go into a little bit more detail. Color is another variation, and then some of the skin markings that we see. So I already talked about thick versus thin skin. Just to review, thick skin is found on the palms of the hands and the soles of the feet. You will see all 5 layers of the epidermal strata present there. But, interesting, we will see sweat glands, but no hair follicles. And therefore, we won't see sebaceous glands. So no hair follicles or sebaceous glands. Now here's something that you should understand. Sebaceous glands are associated with hair follicles. They actually wrap around the hair follicle, which is deep in the skin. So that means if you don't have a hair follicle, you won't have a sebaceous gland. You can't have one without the other in this scenario. But sweat glands, no problem. So look, you can, it's not cheating. Look at the palms of your hands. You shouldn't see any hair there. All right, palms of your feet, same thing. No hair follicles, no sebaceous glands. But I'm sure some of us know you can get sweaty palms, you can get sweaty feet, so there are sweat glands there. Thin skin pretty much is going to be the skin that covers the majority of your body. Important thing to note, it does not have a stratum lucidum. That is not present. In thin skin, though, we will see sweat glands, and we'll see hair follicles, and therefore we'll have sebaceous glands. Look at your forearms. Look at your legs. Peach fuzz on people's faces. All right, there's hair there, hair follicles, and sebaceous glands. All right, so we'll see that in our thin skin. So this slide shows the difference between the thick skin and the thin skin. So you can see here, here's our epidermis. You can see here's our normal stratum basale, same thing down here. All right, our stratum basale is going to be 1 cell layer thick. Stratum spinosum, all right, it's going to be several cell layers thick. You'll see the same thing over here in thick skin. All right, but once we get up here to the granulosum, all right, you'll see the same thing over here. Here's our stratum granulosum. We're starting to get thin. And then look, there's virtually no stratum corneum. And that's your thin skin. You won't have a stratum lucidum here. But look at the difference here. Look at the stratum corneum on this side. Several cell layers thick. And then you have that small, thin stratum lucidum. You can barely see it over here. All right. So that's the first variation, thick versus thin skin, skin color. A couple things that influence our skin color. Hemoglobin, which is the protein, the oxygen carrying protein in our blood. Melanin, that's what helps to give us our skin tone, darker skin tone. And then keratin, not keratin, excuse me, keratin. Let's talk about each of these. All right. So the hemoglobin, the more oxygenated your blood is, all right, the redder the blood will appear. So you'll see in some folks that are light skinned, okay, if their face looks flush, red, it's especially if they've been exercising, right? That's pretty fairly obvious that blood flow is increased. And so therefore, all right, blood is going to have that bright red color because it has the oxygen. Then the next, all right, then the next skin color variation is going to be melanin. All right, melanin, I talked about this before, made by the melanocytes. And again, all right, the amount is going to vary from person to person. Obviously, if you're out in the sun more, you're going to have more melanin activity. Also, genetics, hereditary plays a role in that. All right. So folks that are albinos, those folks have melanocytes, but they just don't produce the melanin. So like I said, more fair-skinned individuals, right, they'll have the melanocytes, but they just won't be as active as folks that have a darker pigmentation. And then there's keratin. And it's always hard every semester when I have to explain keratin because it reminds me of this guy, Jeff, that I worked with a long time ago at Red Lobster. And this guy, no lie, looked like an Oompa Loompa. And if you've ever seen the original Willy Wonka and the Chocolate Factory, all right, these people in the movie had orange skin. And so this guy, Jeff, had orange skin. And it was because he went and got spray tanned all the time at a really bad place. And so our nickname for him was Oompa Loompa, except he was really tall. He was like 6'3". But what will happen is, and you'll see this from time to time, I mean, not too often, but beta carotene is going to be a pigment that you can get from certain vegetables that is relatively harmless, right? It's biologically inactive, so that's why it's a big ingredient in a lot of spray tan locations. But you can also tell that certain people, all right, with their diets, if they have that yellowish orangish tint, it's because they're consuming certain vegetables that have a high content of keratin in it. Carrots, for example, are going to have a pretty high content. Roxy, knock it off. Okay, so this picture here is just kind of showing you how the melanocyte is sharing the melanin pigment with its neighboring cells. And that's what it does. It's the sharing cell. You'll have to forgive me. My dog is in here and she's acting goofy and she's making me laugh. I apologize. I need to focus on you folks. All right, skin markings. All right, let's talk about a couple things that you may have noticed. One, moles, or what we call a nevus. All right, these are a benign growth of melanocytes, but obviously they're prone to malignancy, all right, if exposed to too much ultraviolet radiation, for example. So these type of skin markings should constantly be monitored. All right, freckles, you'll see these localized areas, all right, found scattered in certain folks, myself included. And those are just areas in which we see an increase of melanocyte activity scattered throughout the skin. Hemangiomas, again, another benign type of tumor, and this involves blood vessels. And it'll cause, all right, this skin discoloration. In a lot of cases, it could be like a bright red coloring. Friction ridges, those are just fingerprints. And you'll see, all right, these friction ridges are going to be these peaks and valleys or these folds that'll appear in the dermis and the epidermis. And I'll get into that a little bit later on. We talk about the friction ridges, all right, but it's the interaction between the two cell layers, or not the two cell layers, but the two layers of your skin there. All right, the epidermis is the outer layer. Let's talk about the dermis, the deeper layer here. All right, the dermis is going to be made up of two sublayers in which we call the papillary layer and the reticular layer. And in the dermis, we are going to see our CT proper, connective tissue proper. And so we'll talk about all the different types of connective tissue that we're going to see there. But if you look here on the slide, look what else you see in the dermis, right? Blood vessels, sweat glands, sebaceous gland. You'll even see muscle tissue, erector pili, sensory nerve endings. So this layer is going to have all 4 tissue types in it. Because remember, our glands are made up of epithelial tissue. Whereas when we were looking at the epidermis, that was epithelial tissue. There's no connective tissue in there. There's no sensory tissue in there or muscle tissue. So the 2 layers of the dermis, the papillary layer and the reticular layer, let's start with the papillary layer. This is going to be the superficial layer. That's predominantly made up of areolar connective tissue. Areolar connective tissue is a loose connective tissue, which means this is highly vascularized. You'll see some blood vessels here. Right. And in the papillary layer, we're going to see these bumps. And we call those dermal papillae. That's what these are. They're bumps. And what we'll see is these bumps. All right, here's the dermis on this side of the bump, but on this side, that's the epidermis. And so where they interact with one another or interlock with one another, those are our epidermal ridges and those dermal papillae, which creates the friction ridges, our fingerprints. That's what we're talking about. And you can see here, all right, the epidermal ridges here, and the dermal papillae down here. Where they interlock with one another, that is responsible for your fingerprints. So the layer below the papillary layer is the reticular layer. This makes up most of our dermis, and it's predominantly, and I've seen this asked on test questions before, it's predominantly made up of dense irregular connective tissue. Now, dense irregular connective tissue is, yes, it is a dense connective tissue. It's not going to be as tightly packed as dense regular connective tissue. We're going to have a little bit more ground substance. And so therefore, this type of connective tissue is going to have a little bit more vascularity, right, when compared to dense regular connective tissue, which virtually has nothing. All right. So talking about our dermis here, a couple things that we're going to see in our dermis is when we're dealing with the connective tissue is, remember, connective tissue is made up of 3 things: cells, protein fibers, and ground substance. So the protein fibers, collagen and the elastic protein fibers, right, they're going to be responsible for, 1, the resiliency that skin has, but also its elasticity. And we're going to see in areas of the dermis that these collagen and elastic fibers are going to be in these beautiful parallel bundles. All right. And this arrangement, all right, helps with the resistance of stress, especially when it comes in multi-directional areas here. But what's really important about knowing about these parallel bundles is during surgery, especially plastic surgeons. All right, so these parallel bundles, all right, the way that they move from side to side or up and down, all right, we refer to that as lines of cleavage, which basically tells us where the tension is going to lie, all right, in the skin at that area there. So if we have to make incisions, especially if you're going to be a plastic surgeon, you have to make an incision, it is ideal to make an incision parallel to these lines of cleavage. That will allow, all right, healing to occur much faster, and then that will therefore decrease the chances of scar tissue. If we go perpendicular, all right, that could be an issue. All right, it could leave a scar. All right, and all right, it's more likely to disrupt, all right, the orientation, especially of those elastic fibers there. Now you'll see in some situations on folks that they might have what we call stretch marks. And that is when the skin has been stretched beyond its capabilities. The elastic fibers could not handle that, right? Or some of the collagen fibers have torn. All right, the correct term is striae, but you'll see that in some folks, especially in some women after pregnancy, they might have some stretch marks there on the abdomen. Weightlifters, both men and women, if they start to bulk up in certain areas, okay, they can get stretch marks in and around the axillary area. So here you can see a picture of all those lines of cleavage. And that's why it's ideal to know, right, where the orientation is for these lines. So when you have to make an incision, if you can, control and determine if it's up to you, all right, then you want to go parallel and not perpendicular. Now I bring up this layer that you can see here on the screen here, the subcutaneous layer. First of all, we talk about it in the integument chapter only because it's closely associated with the integument, but do not be fooled. Oh, sorry, did not see that. 1 second. Stretch marks don't ever actually go away. Yeah, for the most part, I'll be honest with you, Christina, I have not met anybody that has any success with using products to get rid of, I know it is sad. Yes, because I have several friends, postpartum pregnant mothers, and they still have the stretch marks. So, and there's some men that have actually, after they've had like gastric bypass surgery, they'll have considerable amount of stretch marks involved too, from losing all that weight. But yeah, unfortunately, they are here to stay. With the subcutaneous layer, this is a great true-false question. It is not part of the integument. Do not associate, I shouldn't say don't associate it with the integument, but if you're asked to name the layers of the integument, don't include this. All right, but you do need to know the other names for it, the hypodermis or the superficial fascia. You'll hear it more known as the hypodermis. We'll talk more about superficial fascia when we get into the skeletal muscle chapter, chapter 10. The two types of connective tissue that are present in the subcutaneous layer is going to be areolar. Again, that's that loose connective tissue, and another type of loose connective tissue, adipose tissue. So this layer, all right, because it has those two connective tissues, but primarily because it has adipose tissue in it, is going to protect, provide storage for energy and insulation and even possibly cushioning. And so because these are loose connective tissues, we're going to have a lot of blood vessels there. So therefore, we like to inject drugs into this area because it allows for rapid absorption of whatever it is that we're injecting into our victim, our patient. Just kidding. So we'll see, right, because of the vast amount and number of blood vessels in that area, it's a great injection site. All right, so let's discuss some of the functions of the integument. I will review this in a little bit more detail in lecture. All right, so just some of the finer points here. Protection. All right, our skin is going to help prevent all right, or help to protect us from injury, all right, harmful substances if you have some sort of, I don't want to say dangerous chemical spilled on you, but anything that can cause irritation to some of the underlying structures underneath the skin, prevents infection, right? Keeps out viruses and bacteria for the most part, certain fungi and parasites. Also helps with when we're exposed to extreme temperature changes, right? When we were talking about the negative feedback and thermal regulation. If you're too hot, you increase blood flow to that area to release the heat from the blood. If you're too cold, we move the blood away from the surface there and keep that warm blood near some of our internal, some of the internal organs and structures. Then, of course, ultraviolet radiation, it helps to protect the underlying tissues and organs from the damaging effects of ultraviolet radiation. The epidermis is an amazing barrier to prevent water loss and gain. All right, you've soaked in a tub before, and when you get out of the tub, your fingers and your feet might be wrinkly, and that's because that tissue is not waterproof. If it was waterproof, you wouldn't be wrinkly. Okay, but it's water resistant, so it prevents the entrance of a lot of water from getting in, but not all of it. And so, of course, we'll see from time to time your body is able to get rid of water through perspiration, through the sweat, and this process of transpiration. And that's when your sweat glands just give off a little bit of sweat and water, right, from time to time. We'll talk about the metabolic regulation in a little bit more detail, I think in chapter 7, in which we're talking about vitamin D or calcitriol. And it actually, the precursor, right, cholecalciferol, is produced in your skin. And then eventually it's got to go through all these different processes throughout the body in which it gets converted into calcitriol, all right, which is a hormone that helps to increase the absorption of calcium and phosphate from your kidneys, and it'll help to release calcium and phosphate from your bones when you have low blood calcium levels. Secretion and absorption, again, when you are exercising and sweating, you're going to secrete certain sweat glands. We're going to get rid of some of their secretions via that way. For those folks that have been trying to quit smoking and they want to use the patch, they're actually taking advantage of the Integument and how well it's able to absorb certain types of chemicals or drugs, right, during transdermal administration, like the patch or some pain patches. My dog got hit by a car. This is a long time ago. Didn't die. But after his surgery, they put a fentanyl patch on him. And so he was nice and drugged. And that was through transdermal administration. Immune function, we talked about the dendritic cells. Okay, the dendritic cells hang out there in the epidermis, but also we do have some of our white blood cells that hang out in the dermis there. Temperature regulation, we discussed, right? When we're dealing with, all right, the loss of heat or the retention of heat, right? So when there's a situation in which you are cold, the blood vessels in your skin are going to undergo what's called vasoconstriction. They're going to get really, really narrow and small. And so they'll prevent blood from flowing through certain areas or decrease it greatly, and it shunts it more towards your core. So it conserves the heat. If you're really hot, those blood vessels undergo vasodilation, and they increase the blood flow there to the surface of your skin, and they'll release the heat. And then, of course, we've got our sensory reception, right? We've got tons of different types of sensory receptors that are going to be found in our dermis there. And we talked about the Merkel cells, right? Those tactile cells that sit up in the epidermis there. All right, we're going to kind of switch over to nails now. All right, nails are a derivative of the skin. So hold on tight. This might get a little bit boring, but just bear with me. All right, we're going to talk about a couple of the structures for these nails. You know what, to be honest with you, I'm not going to do that tonight. I'm going to do that next class. I do want to do just a little bit of labeling here. So this is a good place to take a break, I think, from going over, because I know that if you're not bored by now, you will be if we go over that. So let's do this. Let's pull, if you have your atlas close by, go ahead and pull out the atlas. We'll do some labeling just for a few minutes. And yeah, because even though I can't see any of you, I kind of have a feeling I might be losing some of you. All right, so let's talk about the integument here. So this model is showing us the difference between thick and thin skin. So you can see the bracket there showing you, all right, the layers or the strata of the epidermis. And on one side here, you can see you've got this thing that's supposed to be hair. So that in thin skin, you have hair. Thick skin, we don't have any hair. But we have all 5 layers. We'll go through those individual layers here in a second. All right. On my slide, this looks white. I know sometimes in the lab atlas, it will look yellow. But this right here is that stratum lucida. That's the strata that's only present in thick skin. So it's represented by a white line on my model, but it's probably yellow in your book there. All right, the outermost stratum layer is the stratum corneum. You can see it is several cell layers thick in the thick skin there. And if you look to the left of your screen, you can see it only has a few cell layers thick in the thin skin. Then just deep to that in the thick skin is the stratum lucidum. That's that white line, possibly yellow in your lab atlas. Then you can see the stratum granulosum. This is where keratinization starts to take place. All right, about 2 to 5 layers thick. All right, and then deep to that is the stratum spinosum. That's where we're going to find our dendritic cells. We'll also see the daughter cells in that area there. And then the deepest layer of the epidermis, 1 cell layer thick, that's the stratum basale.
Hey, Dr. Cos.
Yes.
I don't think your screen is sharing with us.
Oh my gosh. Thank you so much for letting me know that. Shame on me.
Yeah.
Shame on me. Horrible, horrible man. All right, let me backtrack then. Okay, real fast. Ooh. Okay. Thank you for telling me that. Okay. All right, real quick. Okay, and where am I going? All right, I will work on trying to get rid of that clicking. I hate that. All right, so here's our skin model here. All right, again, you can see here is the epidermis, the difference between the thick and thin. All right, so we'll start off here. We're going to be pretty much going through the thick epidermis model portion here. So the outermost layer, that's the stratum corneum. Right below it is going to be the stratum lucidum. It's represented by that white/yellow line in your lab atlas. Then below that is the stratum granulosum. Then deeper to that is the stratum spinosum. All right, predominantly looks like, I think it looks, on my screen, it looks brown. And then the dark brown or possibly black is the stratum basale. And that's the deepest layer there. Okay, Now into our deeper layer of the integuments, the dermis. And the dermis is made up of, this is where all the stuff is. This is what I tell folks all the time. The dermis is where all the stuff is. Okay, so you're going to find a bunch of the other types of tissues in this layer. And we have two layers when we're dealing with the dermis. All right, here you can see, all right, on this model, we have a bunch of stuff all throughout. So the more superficial layer is the papillary layer. You can see it's not too big, but it's in close approximation to the epidermis. So in the papillary layer, you have those little bump-like structures there. Below the papillary layer is the reticular layer. This is the larger of the two layers. We'll see quite a few things in there, and we'll go through that. So here's the dermal papillae. Those are the little bumps that you can see. And they'll interlock with the epidermal ridges. This is going to be one of the two structures that helps to form your fingerprints. And usually in these dermal papillae, it's tough to see. I'll bring it over here. Okay, so these little bumps, these are the dermal papillae. And you can see on some of the models, you'll see this little structure here. All right, and we'll talk about this in a moment, but these are nerve endings. They're sensory receptor endings. All right, then you can see these little red and blue things. Those are just blood vessels that were cut. Okay, but in these papillary, these dermal papillae, these little bumps here, you're going to see tons of blood vessels, and you're going to see some sensory tissue in there. Because remember, epithelial tissue is avascular. There's no blood vessels in any of these layers here. So that means these cells have to get their nutrients from the blood vessels down here. And it does so through what we talked about tonight, diffusion. Okay, nutrients and waste will diffuse across into and out of these layers here. Okay, so a great test question would be if you were to cut your finger with a piece of paper, aka paper cut, right? And if it does not bleed, then what layer, or layers, I should say, well, actually, make it easy. What layer of your skin did you cut through? And the answer would be the epidermis, because the epidermis is avascular. So if you give yourself a paper cut and no blood comes of it, that means that you cut deep enough into the epidermis, but not too deep where you got down into the dermis. That's a great test question. All right, well, hold on. Okay. So then again, I have to point this out. Below the dermis is going to be the subcutaneous layer, or the hypodermis. And the two main types of connective tissue are going to be areolar connective tissue and adipose tissue. In fact, those yellow clumps down there, that's all adipose tissue. That's what you see. Okay, moving on. Back to that papillary layer there. Here's your Meissner's corpuscle or tactile corpuscle. That is going to be for light touch because it's close to the surface of your skin. I'll show you it on this other model here. Whoops. Hold on, I'm jumping the gun. All right, so that's the Meissner's corpuscle or the tactile corpuscle. And then you can see this arrow should be over here. All right, these are free nerve endings. All right, and free nerve endings, they play a big role in pain reception. So you'll have some of those just kind of grafting up into there. All right, there you can see the Meissner's corpuscle or the tactile corpuscle on this model. All right, and then in the deeper portion of your skin, you have the Pacinian corpuscle or the lamellated corpuscle. And we call it lamellated because it looks like an onion, much like an ogre, right? It has layers, okay? And you can see here, right, it's deeper down. So this type of receptor is going to be for deep sensations, pressure. All right, the only muscle that you have to know on your first exam, all right, besides the diaphragm, is the erector pili muscle. This is a this is a smooth muscle, right? That means it has no striations and it's directly attached onto the hair follicle here. This is the muscle that's responsible for giving you goosebumps. All right, we've talked about some glands. So now let's talk about the apocrine gland. These are the glands, all right, that are going to be where the tip of the cell busts off. All right, this is going to be a gland in which it doesn't become active. And I'll get into more detail about this later on when we get into tissues, but this is the gland that is going to be, oh yeah, wait, actually, I take that back. I have not gone over this stuff with you yet, all right? Because we haven't, I briefly mentioned it when we were doing tissues, but we'll talk more about it in lecture. I apologize. I'm confusing you with another section. All right. But for right now, just know this is one of the sweat glands. Okay. And apocrine glands are going to be associated with hair. You'll find them in areas like your armpits, the axillary area, and the anus. I'm missing another spot. Around the nipples, all right? The apocrine glands will be present there. The other type of gland is the merocrine gland. These are the common sweat glands, you know, so when you're working outside and sweat just starts pouring down and you get some into your mouth and it tastes salty, well, that's to the gratitude and the debt of the merocrine glands. And so these glands will not be associated with hair. And then the last type of gland is the sebaceous gland. This is the one that produces that oily, viscous type of secretion that you'll notice on somebody if they haven't taken a shower in like 4 or 5 days and their hair looks greasy. Well, that's because of the sebaceous gland here. And they're associated right with the hair follicles. They glom onto the hair follicle and they secrete their secretions, their oily secretions right onto the hair itself. Helps to protect it, helps to kill off microbes, that sort of thing. So speaking of hair, if you're looking at a model and the arrow is pointing to the hair and it's above the surface of the skin, we call it the hair shaft. So in order for you to get it correct, and the arrow is pointing to something that's above the surface of the skin, you have to call it a hair shaft because we call it something different when it's below the surface of the skin. Okay, we call it a hair root. Above the skin, hair shaft, below, hair root. And then at the bottom of our hair structure, where it gets fat, like if you've ever seen tulips, they got a bulb at the bottom of them. Well, here at the bottom of our hair shaft, or excuse me, our hair root, is the hair bulb. And we're going to talk about the different parts of it, but this is kind of like where the hair kind of expands out a little bit. All right, so at the center there, where those blood vessels are, that is called the hair papilla. Blood vessels are going to contain the hair papilla. Just immediately above the hair papilla is the hair matrix. And this is the actively growing portion of the hair. The cells here are dividing and they keep pushing the hair towards the surface of the skin. So where we see that active cell division going on, that's the hair matrix. Now, I want you to think of the hair follicle as the outer coating or covering of our hair, much like an envelope is to a letter. Okay, the hair follicle surrounds the hair. So this black line with the yellow highlight is representing the outer coating. That's the hair follicle. You can see the hair follicle in this model here. I don't know if that looks purple or gray to you, okay, but that's the outer coating of our hair structure. That's what the erector pili muscle attaches onto. That's what that sebaceous gland is wrapping around. That's the hair follicle. And that, my friends, is all of our slides that you have to know for the skin. There's not a lot. There is not a lot. So, questions about anything?